Hello, in this video we will look at the taste reflex and taste cell depolarization, and we will also understand basically the gustatory pathway and sensation of taste. So the taste, for example, the bitter or salty or sour or umami is the different type of taste which that is detected by our brain. So let's draw here is the cross section of the brain as well as midbrain and pons and medulla. So here is the structure of the cross section of the brain stem and brain midbrain. So here is the primary gustatory cortex is here, and here is the left ventral posteromedial VPM is the nucleus of the thalamus. So here is the fourth ventricle, while here is the third ventricle showing in the blue color. So here is the midbrain, and here is the pons, and here is the medulla. So the fourth ventricle are present in the medulla, and the that is the fourth ventricle which that is go into the pons. So here is the primary pyramidal tract is here, which they use for the reflex arc. Basically we will not understand about that, but here is we will focus on the right gustatory nucleus, which that is contain a three nucleus basically. So here is the person which that is contain a pharynx as well as the tongue. So this tongue is basically the three branches of neuron, which that is the geniculate ganglion are present in the facial. So this is the cranial nerve 7, and the petrosal is the cranial nerve 9, and here is the nodos ganglion, which that is a cranial nerves 10 vagus nerves. This is the afferent nerves, which that is carry the signal from the tongue by the sensation of taste, and here is the pharynx. So pharynx is attached with the vagus is attached with. So here is the primary gustatory cortex, and here is the solitariothalamic tract, which that is ascending tract to give the signal toward the primary gustatory cortex. So important thing is that basically it is the afferent neuron which that is carry the sensation of taste toward the brain. While here is I am drawing again the structure of the cross section of brain, which that is we will understand the descending pathway. That was the ascending pathway, but here is we will discuss about the descending pathway, which that is go down to the gustatory nucleus to the for secretion, for the secretion of the something for digestion of food, for example. So here is the efferent nerve, that was the afferent nerve, here is the efferent nerve, and is the solitariothalamic tract and gustatory nucleus. So here is the when bind with the neurotransmitter release into the gland of the salivary gland to produce maxillary gland and mandibular gland, which that as well as the parotid gland, which that secrete of saliva from salivary glands, and amylase, intrinsic factor, sodium bicarbonate, and water. And this is the basically low the pH, acidic to basic. So in this way the amylase enzyme is used for the digestion of starch, so that is why the more taste will be felt, will be sense. So here is the this is the taste bud. This is the taste bud contain a different type of cell. For example, three cells we can classify. So here is three type of cell on the present in the upper taste bud, which that is showing in the figure for sensation of something. For example, the bitter, acidic, or sugary. So this is the type 1, which that is the taste receptor cells containing taste receptor, which that is used for the low salt quantity. While on the other hand, the type 2 is the basically the taste receptor cells as well as the G couple protein receptor. and the taste receptor cells in the form of type 3, which that is a low salt, and the sweet, bitter and sour on the basis of type 3 is used for the sour, and the type 2 is used for the sweet and bitter. So let's draw the whole cells for understanding the mechanism for the depolarization of the cell to give the signal toward the primary gustatory cortex, for example for the sensation of taste. So let's, here is the cell, basically here is the cell type 1 receptor cell, which that is detect the salt. So the low quantity of the salt or low level of the salt when we will eat, that can be felt, that can be uh, sensed. So here is, this is the basically here is, you can see in this diagram to understand, this is the cell, cross section of the cell, which that is the ENaC channel are present. ENaC mean the ENaC is the sodium channel, which that is allowed to sodium move inside when the intact of the sodium chloride, which that is dissociate in the form of sodium and chlorine chloride ion. So the sodium will move inside, so the depolarization will be occur. That depolarization means the inside was negative, but it will become a positive, and outside if in positive, it means the negative conversion. So after the depolarization will stimulated by the ENaC channel to the calcium voltage gated calcium channel will be open to move calcium inside, and again this is the basically more depolarization to stimulate the neurotransmitter to release, is it is the exocytosis. After this exocytosis, this work is done in the millisecond. So binding with the neurotransmitter acetylcholine bind with the neurotransmitter receptor to the create action potential by the exchange of the sodium and potassium ion, as well as you can see in this diagram, and this nerve impulse will move toward the brain from the uh, gustatory nucleus. So here is, you can see here is, we will discuss about the sweet taste. So this is involved in the taste receptor 2 and receptor 1, which that is basically here is the sweet, for example, so sugar, ribose sugar, or galactose, or on the other hand the sucrose, which that is after the sweet receptor heterodimer in the form of here, which that is attached with the G couple protein receptor, and taste receptor 2 and TA TSIR2 and TSIR3, which that is bind the glucose. Glucose is in the form of ligand, and the SEMA AZ, SEMA ZA, which that is bind to detached and stimulate the uh, uh, basically detachment to the gusteucin. Gusteucin is the basically is the alpha subunit like, and will be detached. After the detachment of the gusteucin, basically will phosphorylase, basically GDP convert into the GTP. After this GTP will move into toward the adenylate cyclase, which that is present in the plasma membrane. Basically, the here you can see in this diagram. And after this, adenylate cyclase will change the GUST uh, GUST ATP, GTP convert into the cyclic AMP by the using of ATP, and this cyclic AMP will be stimulate the protein kinase inactive to activate, and this will be bind with the phosphate phosphorylase the channel of the potassium voltage gated channel, which that is inhibited by this activity. So this here is the calcium will be stimulated by this mechanism. Voltage gated calcium channel will be open to calcium move inside for depolarization. After the depolarization will stimulate the exocytosis of the neurotransmitter, and after this neurotransmitter will bind here with the afferent nerves. Afferent nerves bind with the to create a action potential. This action potential will travel into the nerve is known as the nerve impulse. So here is the synapse. This synapse is the uh, uh, taste cells at neuro junction. So the taste cell neuro junction is present here. So let's draw a sour acidic, a sour, basically the acidic. So here is the hydrochloric acid or other acid when we will eat acidic food, which that is or ascorbic acid containing food, which that is we feel the sour. So here is the sour means the proton will be dissociate from the hydrochloric acid, for example, which inhibit the potassium channel, and the depolarization will again that activity create the influx of the calcium, and influx of the calcium which that is stimulate the exocytosis of the neurotransmitter. So what that means, it is afferent arteriole, afferent nerve, sorry. So what that means, that means the different intensity of the action potential will lead to give the sensation of something. So their action potential frequency is not similar but different. So here is the bitter taste or receptor, which that is involved the taste receptor 3 and taste IR3 and 4. In this way, here is the transducin are present in the form of G couple protein. So the G couple protein, the ligand will be bound, and that stimulate the, basically this mechanism is very hard to understand basically. So this is the transducin will be separate as a alpha uh, monomer. So this will go into the phospholipase C, and phospholipase C will be convert the diacyl glycerol. This diacyl glycerol is present in the plasma membrane, and that is DAG will be stimulate the inositol diphosphate into the inositol triphosphate IP3 from the IP2. This IP3 will stimulate the calcium to outflux from the endoplasmic reticulum to exocytose the neurotransmitter. So in this way, this is the four type of taste receptor which that is play important role differently. So thanks for watching. Please make sure to subscribe.